Welcome to this biochemistry lecture on enzymes. So enzymes, as we've mentioned before, are catalysts, and they basically increase the reaction rate without being used up. So they basically, if I like to think of them as something like Pac-Man, that they just sit there and they do their job over and over and over. They don't get used up. Enzymes essentially are globular proteins. There are a few other examples of that, but they're not important for what we're going to discuss. Basically, there's some small RNAs that are also ribosomal RNAs, as you already know, that are enzymes, but that's not exactly what we're going to be discussing here. So most enzymes are globular proteins. The study of enzymes is probably the oldest field of biochemistry. It dates all the way back to the 1700s, and that's probably why we know so much about the field. And it continues to be that way. Reason is because there's some 3,000 enzymes in the human body. Enzymes do the work in cells. They build things and they tear things down. So they're involved in metabolic processes that are really, really important. And the problem is if even one enzyme is defective or missing, there's some enzymes that you're basically just going to die. That organism is not going to function and it's not going to be able to survive. That's why enzymes are very, very important. And enzymes serve as a major source of disease amongst all organisms. Some of the terms that you're going to need to be aware of. There's an apoenzyme, that's the part of the protein, that's the protein part of the enzyme that does the most of the work. There's a cofactor, is the non-protein portion of the enzyme, and it's necessary to catalyze the function. Some examples are a lot of your metallic ions like zinc and magnesium. So they come into the active site and they facilitate a reaction. We also have coenzymes. Coenzymes are, again, non-protein organic molecules. Examples would be some of your vitamin Bs. They act as cofactors for the enzyme. You also have substrate as a term. That's what comes in to and gets reactive, that gets changed. So it's a compound or compound where the reaction occurs. That's the enzyme. That's the molecule that the enzyme catalyzes. And lastly, there's the active site. And the active site is a specific portion of the enzyme where the substrate binds during the reaction. So it's what the part on the protein where the work gets done. Holoenzyme also is the whole substrate cofactor complex. So those are the important terms for enzymes. So let's talk a little bit about enzymes. Enzymes are very, very specific. And that's a very important thing because there's a lot of molecules that are floating around that look and have the same feel as the drug or the enzyme or the substrate that you're going to be reacting with. And enzymes have very high degree of specificity, which avoids side products and byproducts for the cell. It also allows for a capacity for regulation, and it also creates an opportunity for higher reaction rates because these things are highly specific, so they're going to be doing work on a specific molecule. So the example here is phenylalanine. Phenylalanine, it does this in the phenylalanine hydroxylase, which basically is going to add on the hydroxyl group and create tyrosine. If you get a D-alanine, you get no binding. If you get an additional hydroxyl group on the benzene ring of phenylalanine, you get binding, which you get no reaction. So, on tyrosine, if you get additional hydroxyl group on tyrosine, you get binding, but you get no reaction. Again, enzymes are highly specific for what that job they perform. There are 6 classes of enzymes. They're defined by the reactions that they catalyze. There's a couple different ways of naming things, and we'll go over them right now. There's the trivial name. I think you'll hear these, for example, generally speaking. So things like trypsin, thrombin, pepsin, Those are the trivial names. The name gives no idea of the reaction that's being catalyzed or the function of the enzyme. It just is a very common name. So people will use this fairly loosely. There's a systematic name, and that's according to the International Union of Biochemistry, and that has two parts. First, we're going to name it. First part is the name of the substrate. The second part is the type of reaction it's catalyzed. There's a ton of examples of this: phenylalanine hydroxylase, lactate dehydrogenase. Those are very examples of what, or acid phosphatase. Those are examples of enzymes and what they catalyze. There are 6 classes. We'll go over those in a little bit of detail in the next little bit. There's an oxoreductase, in which the oxygen and hydrogen are gained or lost. There's a transferase, where we transfer a functional group, such as amino acid or acetyl group or a phosphate group, from one molecule to another. There's a hydrolase, basically where we add water. This is very common in your digestive enzymes. Lyase, where we remove groups of atoms without hydrolysis. There's an isomerase, where we get a rearrangement of atoms within a molecule. A ligase, where we join 2 molecules together, and you usually use energy that involves the breakdown of ATP. Some examples on the left show the numbering system that also exists. And so there's also an extensive numbering system that the EC has. So the classes correspond with the first number. So EC1 means that that's an octoreductase, and then they break it down further into subgroups. So EC1 means it's oxoreductase, and then there's numerous other subgroups that are associated, and we break those down. We don't need to know those for this class. You notice that the undecaprynyl pyrophosphate synthase is a transferase, so it's an EC2, and then they got the numbering after that. Haloalkene dehydrogenase is an EC3, so it's a hydrolase. Chondroitinase is an EC4, that's a lyase. You can see, hopefully you're getting the sense of what this is and how this works. Again, then the classification is just for your reference. We don't need to really memorize any of those examples. Here's a handy little chart, and it kind of gives you a picture of what is going on in the reactions that we're discussing. Again, octoreductase catalyzes the transfer of a hydrogen or oxygen or electrons from one substrate to another. They're commonly referred to as oxidases or dehydrogenases or reductases. And since these are reduction reactions, an electron donor acceptor is also going to be involved in the reaction. They catalyze group transfer reactions, except for the oxoreductase, which is its own EC. EC2 is a transferase that catalyzes group transfer reactions, excluding the oxoreductase, and they're just basically going to transfer functional groups from one molecule to another again. Hydrolases catalyze hydraulic reactions. These are lipases, esterases, nitrolases, peptidases, and proteases. This is a very, very important class. It's probably one of the more common ones you will see, because we're going to do a ton of protease discussions in the rest of the video series. Lyases, EC4, catalyze non-hydrolytic removal of a functional group from a substrate. It often creates a double bond in the product, and does include decarboxylases and aldylases in the removal direction, and synthases in the addition direction. So they're very, very important. And the picture gives kind of a nice little description of what they are. EC5 is isomerases. They catalyze isomerization reaction, including cis-trans isomerization and some other important reaction. And lastly, ligases obviously catalyze addition of 2 molecules together in order to get one. It is commonly coupled with the breakdown of ATP. Those are the important classifications. One of the this is an enzyme, and you'll notice the reason I'm putting this up there, the binding of a substrate to an enzyme at the active site is the important part of what's going on in an enzyme. And what you're getting there is a very deep pocket commonly, and then we're going to start talking about mechanisms of action and kinetics as we go. But you'll notice that substrate fits in that pocket, and there's key amino acid residues that are going to act on that substrate. And That's the important part of what an enzyme substrate is going to do. And in this situation, this is chymotrypsin, and you can see that we're going to have an amino acid residue that's going to be in there. We're going to cleave uh, an amino acid chain into parts, and that there's key residues. In fact, we're going to talk about in mechanism of action. Usually there's 2 or 3 common, very key residues at the active site that are important on doing the work that an enzyme. Thank you for listening, and we will look forward to seeing you in the next round.